All right, let me keep on going then. Okay, so I wanted to very briefly cover the common rule, which is the federal policy that really guides all ethical conduct of human subjects research in the United States. And there are three major components of the common rule. The first is how institutions can be compliant with federal guidelines. The second component is what makes up an institutional review board, which is the group that reviews research studies in human subjects. And then the final piece, which is what is really critical to the decisions we had to make, is the requirement for obtaining and documenting informed consent. And informed consent is a process that we use in human subjects research where we consent individuals to participate in our study. And to be able to consent somebody to be in a study, we need to do a couple of assessments first. We need to first make sure that they're able to make decisions. So they both have the mental capacity to make decisions, and they're also of the appropriate age to make those decisions. Some of the types of people that would not be able to make their own decisions are people who have uncontrolled mental illness, and also people who are under the influence of substances, and then young children. So those people don't make their own decisions. For the young children, other people, usually their parents or guardians, make the decisions for them. For people who have other issues, we make sort of a determination. But we have to make sure that the person who makes the decision is able to speak for themselves. When we document informed consent for a study, we provide information about the study. We talk about the risks of a study and the benefits of the study. And then we present to the participant the alternatives to doing the study. So if you don't participate, you will still receive your standard medical care, for example. And then we document. And the way that this is documented is usually by a signature that's placed on a consent form by both the individual and then also by the researcher. And then the individual is often provided back a copy of their consent form so that they have it for the records. And then we also keep a copy for our records. So those are the components of informed consent that are governed by the common rule. There are specific situations where you do not need to obtain informed consent to be able to work with human samples. And this is called a waiver. So when you get a waiver of informed consent, that means you do not have to go through this three-step process. The determining capacity, providing the information, and the documentation, that does not have to happen, but you can do research on the samples that you have from humans. And in many times we use what's called a Hipaa waiver, and this becomes very important in the studies that I'll be describing to you. So what is Hipaa? Hipaa is the Health Insurance Portability and Accountability Act. And when you go see your doctor, often you have to sign that Hipaa form. And it's a federal law that requires creation of national standards to protect sensitive patient health information from being disclosed without the patient's consent or knowledge. So we can't hand over sensitive patient data to other groups without their consent. And so most of the time this applies, and this really applies for people who are coming into the hospital or to the clinic and they're getting their routine medical care. A Hipaa waiver is something that we obtain to be able to do research with samples that come from humans, that come from the hospital or the clinic where they went in for their routine clinical care. This is very, very wordy, but essentially I highlighted the points that are important when you think about when you're doing research with samples from humans, the pieces that have to be in place for you to be able to say, okay, I don't need to get informed consent from this participant. I don't need to explain everything to them and have them sign that form for me to be able to do research with their samples. And so these are the criteria you have to meet for you to be able to obtain a waiver of informed consent. So there needs to be no more than minimal risk to the privacy of individuals. And then you have to have a plan. You have to have a plan to protect the identifiers from improper use and disclosure. You need to have a plan to destroy the identifiers at the earliest opportunity. So as soon as you don't need the samples anymore, you want to destroy those samples. And you have to provide an assurance that the protected health information will not be reused or disclosed to any other person or entity. Anybody outside of the research group cannot have access to that information. So this is what allows you to do research on samples that are collected as part of routine clinical care. Okay. So that's the basics. I think that should be enough for you to understand what we then went through. And now I'm just going to talk about our study and explain to you what happened along the way. And a lot of these principles will come into play. So the Seattle flu study was started about 3 years ago now. This is the group of investigators that work on the Seattle flu study. We now have over 250 people involved, and it's really a large team effort. So the reason why we developed the Seattle flu study was because we were working to figure out a way to prepare for the next pandemic. So looking back at 1918, we know what happened with the 1918 flu was that there was a world population of 1.8 billion, that the primary mode of transportation were ships and railroads, and it would take about 4 months to circulate the globe. And the preventative measures that we had in place were masks and disinfectants, and then the treatments were bed rest and aspirin, and 2% of the population ended up dying. So we were in a situation where we saw what had happened in 1918, and we felt like what we needed to do was to prepare for what might happen again, given the inevitability at the time in 2018, when we were thinking about this, of another pandemic emerging. So of course, now we're in a pandemic. I would like to say that we're at the end, but I don't think we are. But we are still in the middle of a pandemic. The world population is quadrupled. Now people use airplanes to get around, and it takes about 2 days for viruses to circulate the globe. Fortunately, along with the masks and disinfectants, we have vaccines. And we also have good antiviral drugs. Nevertheless, we have about 4 million people who have died due to coronavirus, and many more who will die in the next several months as the new variants take over. So back when we started the Seattle flu study, it had been 100 years. We felt that another pandemic was inevitable, and we felt that the strategies that were in place in the United States were really not designed to detect novel pathogens. So when we set up the flu study, it was to develop a pandemic surveillance platform to find new pathogens and to contain spread before the months to years that it would take for a vaccine to be developed. And we called it the Seattle flu study because we used flu as our pathogen, and also because we felt that flu would be the source of the next pandemic. So what we knew was in place at the time, back in 2018, and continues actually to be what's in place now, is that people come, so we have that the existing surveillance strategies really relied on people coming in for care. So they needed to essentially come into the hospital for symptoms of a respiratory illness. And then the providers would then collect a swab from them. And then those swabs would then be sent to the state public health labs, and then they would be tested for flu, and then some of them would go on to further testing. It really did not do a good job, and continues to actually not do a good job of capturing people outside of clinical care settings. So if somebody in the community, like a young adult, a college student, for example, or a 30-year-old who has young kids at home, they can get a cold, they cannot get that sick, and they probably would not go in for medical care for a runny nose. And so they would then be missed by the systems that we have in place. And so our hypothesis was really that it is those people, those young, healthy people in the community, who could be the ones who could serve as the early signal of new viruses that are emerging. Because they're going to be the ones who are mobile, they're going to be the ones that travel, and they're the ones who will bring it in. By the time it shows up in the hospitals, in the young children and in the older adults, we will have missed a couple of weeks of time, or we would have been able to capture it early had we looked in the community. So what we wanted to do was to then test this, to see if we could figure out a way to enroll people who are not necessarily seeking care, but who are in the community. So we expanded testing to the community. We set up kiosks at a couple of places around the city of Seattle, on the University of Washington campus, and a couple of childcare facilities and schools, workplaces, homeless shelters, transit stations. Leah Starita, who is a faculty member in genome sciences, also simultaneously built up a high throughput lab that could simultaneously identify multiple viruses, and also developed a way to test for new pathogens very quickly, and then to do whole genome sequencing to map chains of transmission, which was then mapped out by Trevor Bedford's group at the Hutch. We wanted to be able to establish a way to take all of the data that was coming in, both clinically from these front-end kiosks that we had set up all over the city, and then also merge that with the laboratory data that was coming in from Leah and Trevor to be able to figure out things like who is getting what pathogen first, and then where does it start and where does it spread. And then ultimately, we also wanted to look at new ways to diagnose and treat pathogens at home, because we felt like the best thing to do was to be able to diagnose it and treat it early enough so that people then didn't have to come into a clinic or a hospital and then potentially expose others. So that first year we started in 2018, we enrolled at many, many sites across the city of Seattle. This is a map of, or this is a sort of a snapshot of all of the different local partners that worked with us and allowed us to work in the community, to enroll at public schools, homeless shelters, all of these different places. We were in the international arrival zone of SeaTac. We were in Hutch Kids. We were in all sorts of places where people who are normal, healthy, would not be seeking care necessarily, but could be carrying respiratory viruses, were intermingling in the community. Simultaneous to this, we also collected samples from the hospital. So we brought in samples that were collected at the UW hospitals and at Seattle Children's Hospital, along with their associated clinical data. And then we pulled them in through the same platform that we did the community enrollments. And really the goal was to see, was there any difference? Were the same pathogens showing up in the community as in the hospital? And were there differences in both sort of the type of people who were getting the viruses in these different places? And then also, what was the pattern of spread? Did it start first in the community and then go to the hospital? Did they all show up simultaneously? So those were the goals with these two types of enrollments that we did. And then we then took the samples that came in from these kiosks. We enrolled them as shown here. We screened them and then we enrolled them. We collected a swab from their nose. We gave them a gift card for participation, and then we took it back to the lab. And then all of these samples then were fed into this platform to be able to simultaneously look for multiple pathogens. One of the other things that we developed was to have a way for people to enroll from home, and we called this Swab Send. And this was really a way for people who were sick and not coming into the hospital or the clinic and not going to work and not out and about in the community to be able to go to our website and to go through an eligibility questionnaire and then enroll in our study and then have a swab kit sent to their house within about 2 hours. And then they would then swab themselves and then send it back to us at the lab where it would be tested. So this is the what we call a quick start guide. It's what we sent home with them when they called or when they went to the website and told us they were sick, and it gave them instructions for how to self-collect a nasal swab. At the time, this was not standard practice. It is what is permissible now, but at the time, self-collection of a nasal swab at home for respiratory viral testing was not standard practice, and is still, debated about whether or not this is an approach that allows us to be able to find viruses early. I think it is, and I think many others think it is as well. So then all of these samples that come in from the clinics, from home, from the Swab and Send process, and then from the hospitals and from the kiosks, from all of these different places are then fed into Leah Starita's lab, where she puts them through this multiplex assay, and we simultaneously look for many, many pathogens. And then the ones that were flu positive were then moved on to whole genome sequencing with Trevor's group. Again, this is just showing the pathway. The samples come in, they get extracted. We do detection, a multiplex detection method for 27 pathogens, and then the ones that were flu positive, and then subsequently SARS-CoV-2 positive were then moved on to the whole genome sequencing. And the key with all of this is that, first of all, it was very fast from the time, even now, from the time of sample arrival in the lab to whole genome sequence is about 1 week. And then also the clinical data that we capture is also simultaneously fed in at the same time. So that if you were to collect a sample and then test it and find something, then you would be able to very quickly go ahead and link back to the information about the individual and then take action based on that. And then one of the other key pieces that we put in place was the ability to be able to see your results. And this was something that I'll talk about a little bit more in the future, in future slides. But one of the things that we really envisioned for this study was the ability to be able to collect a sample, to test it, and then to be able to go onto our website in 24, 48 hours and know whether or not you're positive or negative for the virus. And so for people who are enrolled in Husky coronavirus testing, for example, they get a barcode once they take a swab, and then they just go to our website and put in the barcode, and then the result comes out. This is the results portal. It's again, it's a very easy way to really provide participants back what information about what they submitted. And I think this is the question is, when you're able to provide this information back and tell people you have the flu or you have SARS-CoV-2, how does that impact their behavior? Does it make them less likely to go to work or go to school, or more likely to mask, to stay home? All of those things. Those are questions that we wanted to answer way back in the day before the pandemic. So in that very first year of the study, we enrolled a lot of people. We enrolled over 5,000 people in the community. We enrolled over 12,000 people from these hospital samples. And we found that overall, comparing these two groups, the ones in the community and the ones in the hospital, about 20% each had flu detected. And flu was the pathogen that we were really focused on at the time. And I think this was, for us, this was a success. What we were able to do is to develop a system where we could capture people in the community who had flu. And the number of cases that we found, or the prevalence of community flu, was the same as the prevalence of hospital flu. And I think that shows us that there is flu, undiagnosed flu in the community that's circulating, and that's probably being transmitted by people with mild symptoms or with no symptoms, and probably leading to then the cases in the more vulnerable who then subsequently get hospitalized. We found that when we looked in the community, 39% of people had influenza-like illness, which is what CDC uses to sort of define flu for their studies. That being younger and enrolling in the waiting room of a clinic were more likely to, were predictors of being flu positive. We wanted to know how quickly we could get to people in the community, because the earlier you get to them, the more likely you are to prevent spread. We found that we were able to enroll about a third of people within 72 hours of symptom onset, which is pretty good for this type of study. And then of the adults who enrolled, 21% reported seeking care for their illness. And that also was important to us because it showed to us that 80% were not seeking care for their illness. So these people were walking around in the community, and we collected a swab and found flu, but they weren't ever seen by the care system and would have never been captured through any other method. So the samples were then sequenced, and then we used them to really map out the different strains of flu that were circulating in Seattle that very first season, the 2019 season. And then we also were able to use the data from those samples to then map how the virus was spreading in the community. And so this is a phylogenetic tree on the left, and time is on the x-axis. And on the right is a map of the city of Seattle. And so basically what you're going to see in this animation developed by Trevor Bedford is that first flu enters here, this initial strain was detected in a college student who had recently traveled back, and then basically started spreading throughout the city of Seattle. And different strains would come in, and some would lead to larger clusters, and some of them would extinguish very quickly. Basically started developing a method by which we could potentially map the spread of viruses in a community, and map how an outbreak evolved. So that was the platform that we developed in year one. And we felt like we were pretty successful at our first year. You know, we did what we wanted to do. We developed the infrastructure for community-based detection. We were able to get from sample basically to genome, and then to develop these types of maps as ways to visualize the data, and really felt like this could represent a path forward for community-based pathogen surveillance for pandemics. So then we started year two, and in year two, we wanted to do a couple of other things. One of the things that we really wanted to understand was whether or not we could stop transmission early in congregate settings. And so we started a randomized trial of on-site testing and treatment for flu in homeless shelters in Seattle. We also started a clinical trial of delivery of flu diagnostics and antivirals to the home. So we partnered with a company in Australia called Illum, which actually ended up getting the first EUA for home COVID testing. But they had a home flu test in development, and we provided this to family members of households with young children and asked them to test themselves for flu. And those who tested flu positive were then sent a delivery of a flu antiviral within a few hours to their household. So really home diagnosis and home treatment for flu, with the hope that this could serve as a model for preventing spread during a pandemic. And then critically, and we also decided to expand surveillance for respiratory viruses to households with young children. And this is actually where the first case was discovered. So this surveillance platform that we developed. So, we had just started our second year of the flu study. We were in pretty good shape. And of course, at that point, we started hearing about the cases of SARS-CoV-2 coming out of Wuhan. And Trevor and Mike Famulari at the Institute for Disease Modeling took a look at the data, and they essentially came to the conclusion at the time that it was inevitable that it was going to come to the United States, and it was just a matter of time and being able to find it. So I wanted to sort of just run through the timeline of what happened in those early days, in case others may have forgotten. So this was what was happening nationally. So on January 21st, 2020, the first US patient was identified in Snohomish County in Washington. On January 22nd was when we started discussions with CDC, Washington State Department of Health, and Public Health Seattle-King County, really about potential for taking all of these samples that we had collected as part of the flu study over the past several months to years, and testing them for coronavirus. We felt like this represented a way to be able to know whether or not it was here. If you recall, at that time, the CDC was really the only entity in the nation that was authorized by the FDA to test for coronavirus. And that testing was really only intended for individuals who are travelers and those with severe symptoms. By February 5th, so about 2 weeks later then, FDA authorized an emergency use authorization license to the CDC to produce a diagnostic panel that could be used by local public health labs, so that testing could happen not just in Atlanta, but really at the local labs. For us, it would have been in Shoreline. However, I don't know if you remember, but way back in February, there were lots and lots of issues around contamination and inconclusive results because of issues about how one of the controls was made, which really was not discovered until much later. But because of that, even though there was authorization for CDC to produce these tests to be used by local labs, in essence, there was no testing available because the tests that were available were faulty. So we talked with CDC, FDA, YDOH, Public Health Seattle-King County multiple times for the next several weeks, and sort of came to a final decision that we were permitted to test for SARS-CoV-2 and the samples collected as part of Seattle flu study. And so we started on February 25th, 2020. And as been now well publicized, we found the first positive 2 days later. And that was from a Seattle area teenager up in Snohomish who had no recent travel or other epidemiologic risk factors. And importantly, this sample was collected from the hospital residual study. And if you remember, this hospital residual study is the samples that were collected from the hospital from patients who are coming in for routine clinical care. We then took the sample, we confirmed it was positive on 2 separate other assays, and then we also sequenced it just to be totally sure. And then Trevor posted about this on Twitter, and then it was picked up by the New York Times. So and so what they describe in this article, which is what we found, is that there were 2 cases detected 6 weeks apart in Washington State that were genetically linked. And what it showed was that there was cryptic transmission, that there had been ongoing transmission in Washington State during that 6-week period. And in many ways, this was sort of what allowed, I think, for more alarm to be raised about what was going on. I mean, at that point, there was also the skilled nursing facility outbreaks, a couple of other things, all starting to emerge around the same time. And so it really just did provide an opportunity, though, for us to sort of coalesce around a more urgent national response to coronavirus. We then subsequently published these findings in 2 papers. First, where we described the clinical data showing that that first case indeed was from February 24th. We tested all the way back to December. And then also showing the genomics of the strains that we picked up, and showing that there was evidence of community-based transmission at the time. And so we went back and we tested all of those samples, 2,353 samples from people who enrolled. We found that that 25 of them had COVID-19 detected from those early days, and that only a third reported seeking care for their illness. So I think what it did end up showing us was that we were able to, again, using this type of community-based surveillance approach, find people who were not coming in for care. People who were not necessarily seeking care, but yet had coronavirus, and that people could essentially enroll at home, take a swab at home, send it in, and then we could pick up a novel pathogen. So it was proof that what we had developed for flu could apply to other pathogens. This was some other work that we did. This is actually led by Denise McCulloch, who is who just finished her fellowship in my lab, and she really wanted to answer the question of, when you take this sample from the home in unsupervised way, how concordant is it with just a sample that's collected through routine clinical care, like an MP swab that's stuck up your nose? And she found that these samples were highly concordant, that in fact a home collected swab was 80% sensitive and 98% specific compared to one that was collected in the hospital. And that if you looked at those with higher viral loads or lower CT values, cycle threshold values, that the sensitivity of home swabbing was 95%. So again, sort of demonstrating that home swabbing for these novel pathogens is an effective path forward for pandemic surveillance. And then finally, one of the other things that we found also on further analysis is that people could swab themselves, that this was a study that was led by Ashley Kim, who's a research coordinator in my lab, who really looked at all of the swab kits that went out in that time period to see how many people were able to do it error-free, and then how many people were able to have a pathogen detected in that sample. And this was before COVID. So, but what you could see here is that of the people who returned the swabs, the vast majority of them were testable, and that we actually recovered a lot of virus in these samples. That 34% of the samples that were submitted to us from these home-based swab and send studies were positive for a pathogen. And that it was actually very quick, the way that we deliver the swab to people, that the median delivery time from request was 2.2 hours. So that if you put in your request for a swab to go to your house because you were having symptoms, within 2.2 hours, you would have the swab kit at your house and you would be able to collect the swab. So really a mechanism that could be used for pandemics moving forward. So here we are at a point where, we had found that first positive, we sequenced it, we described community transmission, we demonstrated that this is both safe, error-free, fast, and concordant with provider collected swab. And so what do we do at that point? And so this is where we started discussions with FDA, CDC, Public Health, Human Subjects Review Board, and the Washington State Department of Health about sort of what to do in this type of situation. And so if you remember, there were 2 types of enrollment. There was community enrollment and there was hospital enrollment. And this actually came up there was issue with both of these types of enrollments. So community enrollment, people went through the informed consent process. So they were told about the risks and the benefits of the study. They were given the alternatives and they signed a consent form. However, the issue that came up, and I'll show it to you in the next slide, is that the consent form didn't specifically say we were going to test for SARS-CoV-2. And the reason for that is because when we developed the consent form, when we were doing the study, it wasn't a pathogen that existed. And so we weren't able to write that into our consent form. So our consent form instead read like this: after you consent to participate, enroll in one of our studies and have a sample collected, we will test your sample for the flu and other respiratory pathogens, germs. We may perform further testing to analyze the specific pathogen strain if it is found. We also, through this consent process, told people that Washington state law requires reporting of infectious diseases, including flu. But we didn't explicitly discuss the use of our research infrastructure, including the data or samples, for other public health activities beyond reporting to Washington state. So this was the issue that came up of, when people signed this informed consent and we said, we're going to test for all of these other respiratory pathogens, we did not explicitly say we were going to test for SARS-CoV-2. And so the federal government and lots of other people, had issue with that because they said that essentially we needed to have a process by which we consented people specifically for testing for SARS-CoV-2. And that is how we would be allowed to test for SARS-CoV-2. We argued, of course, that when you write a statement, we will test your sample for flu and other respiratory pathogens, and we may perform further testing, and that the overall goal of the study is a pandemic surveillance platform, and that encompassed testing for pathogens that hadn't yet been discovered, that we couldn't explicitly write into the consent form. And so that became a point of debate, and it became a point of debate for quite a long time of, is this consent form enough to allow us to test for SARS-CoV-2 in these samples, or do we have to go back and reconsent all of these participants to be able to test? Because are we being fully transparent with what we are doing when they sign that consent form? So that was sort of issue number one. The other issue was around the hospital samples. The hospital samples, if you remember from the very beginning, we tested using what's called a Hipaa waiver. So we did not obtain consent from the participants. And so this is fairly standard for research conducted using leftover samples that are originally collected as part of clinical care. So for example, if a person comes into a clinic with a fever and a cough and they have a swab that's collected for flu testing, other researchers can go ahead and obtain a Hipaa waiver and use that swab and test it for a lot of other things. But really the issue that comes up with this is not so much about whether or not we're allowed to test, but whether or not we're allowed to return the results based on this test. And this gets into sort of the complexity of Washington state law and sort of laboratory regulatory guidelines at the time. Just quite complicated and things I really did not understand in a lot of detail as a clinical researcher until all of this came up. And then I became very, very much embroiled in the details. But essentially, we did testing in our lab as a research test because we were doing a research study. We did not have what's called a CLIA licensure, which is a clinically, it's a clinical license. It's basically allowing you to do testing of samples for clinical use, for clinical decision making. So if somebody comes into the hospital and they had a flu test done in a clinical lab, then the provider will get that result and they'll say, okay, they have flu, I will give them a drug for flu. So our test was not designed like that. It was not meant to be able to make decisions on. So really what it came down to for samples was we could test, but we couldn't do anything with the information. We weren't allowed to return the result or report out the result because it wasn't a clinical test. But then the issue was that it was a pandemic pathogen. So that if we found it, we felt, and many others felt, that action had to be taken based on that result. That was sort of the other major discussion that went on for a very, very long time about what do you do in a setting where you are a research group doing research testing and you can't take action on the results of your research test, yet this is a public health emergency. Can we do anything with that information? Can we take action at all? So, so the way that we approached it in the setting was to really ask ourselves the question, was it ethical to withhold results of a test that could potentially alter the course of an individual's illness and society's trajectory, given that all of these caveats, that the testing was done on the sample without informed consent because it was a hospital sample? We have a research result. Can we take action on that research result from that sample, or do we have to withhold that information, keep it to ourselves and not take action? We can use it for just sort of describing potentially in an academic manuscript yes, there's community transmission of SARS-CoV in the United States, or do we go ahead and report that result out to people who would take public health action, such as the provider or local public health? And so that was the decision that we had to make. And so here we are. This is what, so as I said, based on our current research study guidelines, we weren't permitted to return that result back to the participant. However, there were a couple of other things that we had to think about. When we were authorized to start testing on February 27th, the sample that we tested that turned positive was from just 48 hours earlier. So it had just been recently collected, and the viral load was very, very high. It was very high. And based on the information that we had at the time, which was still very early in the pandemic, we felt that the individual was still likely to be symptomatic and at risk to transmit the virus. And we were pretty concerned that if we didn't inform the participant and the clinicians, then we wouldn't be able, then that person potentially would not have been able to be diagnosed for some time. Because if, as you recall, the only test that was available at the time was the faulty diagnostic test that CDC had developed for the state labs. It really wasn't functional. And so we were concerned that this person could then go from place to place, get tested again and again for all sorts of things, and potentially get much sicker, expose others while this was all happening. So we came together as a group of investigators and we sort of had that discussion of, what is the right thing to do in this situation with all the caveats as they laid out. And then we made the decision to go ahead and notify the hospital. And then the hospital then notified the patient. And when we did it, what we knew was that it would represent a breach, an ethical breach, according to what our research study guidelines were. We felt like in the setting of a public health emergency and a pandemic, we decided that was the decision we had to make. So this is actually at the bottom here. It's a paper that was written about our study by somebody I don't know, but it was actually a very accurate representation of what happened. And so, it sort of lays out, and I really just pulled this from the paper, but what happened at the time. So, based on the fact that we then decided to make the decision to go ahead and report, we were then told by CDC and FDA to stop testing. And then we're told that we could add in new consent language. And we really had to say explicitly in our consent that we were testing for SARS-CoV-2 and that we were using these for public health activities. And then we could prospectively, moving forward, not backward, test those samples. And then subsequent to that, we also spoke to our institutional review board at UW, and then who made the determination that given the public health emergency, we did indeed at the time that we made the decision that night, have an ethical obligation to test all the samples. And they said that we already had consent from participants to test for another communicable disease and return results. And therefore we were already engaged in both research and public health, even if it was not explicitly laid out in the consent form that way. So here we are. This was sort of what I wanted to sort of open up the discussion for. These questions that we had, and then also that, we have to think about going forward for other studies. So really sort of the question of during a public health emergency, like a pandemic, how do you distinguish between what is research and what is public health? Public health activities are exempt from the common law, which, as I showed you before, is the informed consent, the IRB, all of that stuff. So if it's a public health emergency, a lot of those things are exempt. And when is it appropriate to repurpose research activities for public health during a pandemic? There are many of these types of biospecimen repositories in place, not just through us. There's a lot of well-curated metadata or clinical data that goes with these samples. Many individuals who enroll in studies consent to have their samples used for other studies. It's called secondary use. It's pretty standard. A lot of people sign that type of consent. Is there a mechanism that we can develop in the future that allows us to be able to leverage these types of platforms and these repositories without having to go through what we went through during this pandemic? So that's really the question that we have as a group of investigators, and sort of looking forward and thinking about where things are and where things are going. we're not really in a place where this would not repeat itself, that not a lot of legislation or not a lot of things have been done to make it easier to potentially take biospecimens and to be able to use them to help the pandemic response for future pandemics. So, so in conclusion, early in a pandemic, we found that it was difficult to convince others that early urgent action was necessary, that biospecimen repositories linked to the clinical data are really quite useful, that community-based studies can provide an opportunity to find pathogens early. The situation that we were in, what did not fit into any existing guidelines at the time, and still doesn't fit into any existing guidelines now. And that I really think that for the future, that if we want to really be responsible and ethical researchers during a pandemic, we need better processes in place to be able to allow us to repurpose, to be able to do things with the samples that we have and the data that we have during public health emergencies that could help the public health response. And I think that is it. I'm happy to take any questions.
Yay, thank you so much, Helen. That was so interesting. A number of questions have come in the question and answer box. So I think we will just go through them in turn. I want to kind of encourage, we have, you know, over 200 people here watching you virtually. I'm sure other people probably have questions. Please, if you have questions for our speaker, please put those in the Q&A box. So the first one comes in from Tim Brown, who asks, how did the Seattle flu study deal with misinformation, disinformation around SARS-CoV-2 and the federal public health response? Were people more hesitant to participate in the study because of worries related to conspiracy theories or disinformation campaigns? What did that look like on your end?
We did not have we didn't have any sort of negative response from the community, actually. We had overwhelming positive response from the community. I think we actually ended up getting more people to participate who wanted to be part of the study after this. So I think it really, you know, I think people who tend to participate in the Seattle flu study are not the people who tend to not believe in testing. So it is, you know, it takes a certain person to want to do this type of research. But we haven't really encountered that very much, fortunately.
Okay, terrific. Another question came in from Andrew, basically saying, I'm confused about the informed consent debates and those arguing to withhold information for participants. Were any participants arguing that they did not want the results shared with them, or is this pure speculation on the part of non-participants?
Yeah, so this comes down to sort of what is allowed to be shared versus what is not allowed to be shared by the regulatory groups. So we are, according to Washington State, and it's sort of debated nationally about whether or not this should be applied nationally, but Washington State has determined that if a test is done in a research setting, that result cannot go back to the participant. And it has to be done in a clinical setting for that result to go back to the participant. So it's not the participants who don't want to know, it's that we are not allowed to release those results to them unless we are a clinically regulated lab. And we are now a clinically regulated lab, which is why we return SARS-CoV-2 results to people.
Mm-hmm. So maybe that might possibly also then be the answer to this other question about why you decided to notify the hospital as opposed to the patient. Was that the similar reasoning there? So you basically could not return to an individual research participant?
We could not. That was part of it. But we also felt like for this particular case, since the sample was collected in the hospital, that it was really the responsibility of Seattle Children's Hospital to decide the path forward. And we didn't want to do that. It turned out that that particular person was already enrolled in one of our studies, and we did have informed consent from them, but we didn't find this out until after the fact. When we made the decision, we did not know that. So we wanted to make sure that we went through the right channels, and this, you know, involved a lot of discussions about how to do it properly, and you know what was the least harmful way to be able to do this, but do it quickly enough that action could be taken. And we did end up being able to get that result back to the participant that very next morning, right before he returned to high school. So we were able to prevent an exposure of an entire high school.
Right. There's a question from Hannah. Did you face any consequences for the violation where you returned the result to the hospital, other than that they asked you to stop testing? Did you expect other consequences? And I mean, Helen, as you know, when we invited you to come and present to us today, I mean, in research ethics, we talk sometimes very broadly about doing the right thing. It's hard to do the right thing. And I think that's what Hannah's asking about here. Could you talk to us a little bit about that, what that was like for you as an investigator leading a study of this kind and in that situation?
It was, there were consequences, certainly. There continue to be consequences for this. I think, currently under FDA review for the study. But I mean, I think ultimately you have to sort of decide at that moment in time, because you're not going to know what the consequences are necessarily. Well, you will know the consequences of not notifying, right? And so that was the most immediate urgent thing to us, of what would happen if we didn't let them know. And so the other stuff seemed not quite as important as making sure that we could take action on that sample.
Thank you. So Robert gives you his thanks, as I think many of us do. Then he asks, when you and your colleagues were debating about what to do, who did you feel compelled to involve, and what was that process like? You've named a lot of players here and a lot of conversations. What did that look like? Could you give us a little bit more? I mean, you've already given us a lot of great detail, but also your thoughts on how to navigate some of these ethical tensions that that arose in this context and could arise again in similar research scenarios.
Yeah, we did not involve. So of course this happened, I think, late at night. So we could not at the moment involve people that we would have otherwise involved during the day and did involve the next morning, including compliance and legal and all of those people and human subjects. But that night we've had to make a decision. So we made it as a group of investigators with the involvement of public health. We called public health at local and state levels, and we all made the decision together.
Okay, all right. Anna Mastriani, research ethicist here at the University of Washington, says thank you for your presentation and asks, were all of the samples collected with identifiers that would have allowed for recontact, or were at any point were some of these de-identified? How did you deal with that issue?
Yes, that came up a lot. That's a great question, this identifier question. So at the time of testing, they were all identifiable and were able to link back. I believe subsequently broke the link, but at the request of, I don't remember actually if this is, if I may be misspeaking. I can't remember. We were asked to de-identify, and then we did go ahead and de-identify when we were asked to. But I don't remember what happened, but they were certainly identifiable back to the participant at the time that we did the test. Yeah.
I mean, we have one more question, but I mean, just my own kind of editorializing here. I mean, I think, it's so, this very interesting ethical concern about, like, where does research end and public health begin, right? But you're doing research with the aim of enabling public health surveillance, right? So it gets very, very complicated when you're basically being intentionally asked to do something that would sort of mitigate the overall objective of the study, right? So that seems like to be a very central ethical conundrum here. Anyway, yeah, enough of my comments. Another anonymous attendee, what did the SFS participant demographic look like, socioeconomic, et cetera?
It depends. So we work in a lot of different communities for Swab and Send, in particular, which is sort of where these types of samples came from. It tended to skew more white compared to others. And then it sort of was bimodal in income distribution, like lower and then higher, but not so much in the middle. But overall, we are fairly representative of the community, because we do pull in from, public schools, homeless, a lot of traditionally underserved communities. So, but that particular piece of it tends to skew more white.
Thank you for that. Have you ever, so Sia asks, have you ever felt the consequences of your work have trumped the satisfaction of your work? So I'm not quite sure. If so, how do you or did you deal with that? So consequences versus satisfaction.
I think obviously we didn't know that this would become sort of a New York Times thing, so we didn't. It was at the time it was just a decision that we had to make. Seemed like a very private discussion. And then it became a big deal. So I mean, I think if I were to go back and do it again, I don't think I would do things differently. I think we had to make that choice at that time. I wish we had been able to do it earlier. I wish we had been able to take action faster. But we weren't, and ultimately it's fine. It was a very, very difficult time for, I think, the US. Things were very chaotic at the national level. And I think thank goodness we have good people in state and local public health. So
well, there's a number of questions, remaining questions, but mostly it seems to me that what's coming up now are expressions of gratitude. So I will let you savor those and take those in. We are at the end of our time. Thank you so much for joining us here today. Thank you so much for telling us about this remarkable story and about your courage, your moral courage in the face of this. And anyway, we are very grateful to have you here with us. Very grateful for you to have shared your story. Hope that will be some inspiration for the trainees who are here participating as part of the BRI program. So thank you so much again.
thanks for having me.